First, thank you to the organizers for inviting Inger Skrede, who's sitting there, and me, to present some of our recent work in the Pezizomycete on Helvella. And it should be a short talk, which means that we have to drop all historical data in relation to Helvella. If not, we will go over time. But because I think it's important to give some take-home messages when we do taxonomic work, combining morphotaxonomy and molecular taxonomy and so. And because I know, and I'm seen from the other talks previously, that we have challenges in relation when we want to survey big genera with partly narrow and partly broad species concepts. And here you have an example, and some specific Helvella things will certainly also apply to others who work on taxonomic revisions and in relation to the large body of species. Here you see Helvellas. They are the most elaborate, elusive, large, easy to find if you know where to find them. Outside, they are abundant on the northern hemisphere, in forests, and at higher elevation. And then it's important also that the reason why they haven't been frequently collected in the southern hemisphere and in the tropics, so that's because you have to go up at higher elevations, up in the mountains, and there they certainly occur. So they definitely have a worldwide distribution, and they are nice. So let's go to our work that you can see is also summarized in this abstract. I got this from Karen Hansen yesterday, just to show you where we have, it doesn't work, yeah, it's here. I can see it. Helvellacea, you are down there in the Pezizomycetes, in the B branch, very close to Gyrometra and Ricina and the other tuber and so. And Helvellacea, they don't have an asexual anamorph, just to conclude based on the previous collections. The species recognition is rather problematic in Helvella. Kerry O'Donnell has recently, over the past 5 years, also shown that you have exactly the same situation in the even more famous genus Morcella. And it's important for me to stress that what we've done here in our work is that it's just a beginning of understanding species diversity and species relationships. Usually, taxonomists, they are too eager to encode their results into classification schemes and establishing a lot of intrageneric groups and of course, but that should be postponed, I think. That's a message, until we have got really good phylograms with good support and things that we can call clades in modern phylogenetic classifications. This is a survey of a lot of specimens, 100 years back, the best, some of the dried collections. They are, and look, mainly from Europe, but of course to equilibrate our species concepts. It has been important to see what we think is the same species from another continent. How do they look in relation to the European? So we have included a few additional specimens from North and South America, Asia, and Oceania. I think this slide is the most important one, telling you that we worked really, it's been a real hard work to get to the combination of these secondary barcodes. I mean, if you see those 4 short sequences from LSU, RNA polymerase II, the heat shock protein, not many using that, nothing in the on the databases, and elongation factor. You see that to have amplification and sequencing success in Helvella, they had to be designed as purely new. They are short, and ITS you will miss there because that's a general conclusion. ITS will not work in Helvella. And I could have given you a long lecture on that, but we have to drop it off. We can discuss it afterwards if you have questions. And when we apply maximum likelihood and Bayesian analysis on our set of concatenated sequences and get a concatenated tree like this, the importance is not the backbones, because this is not an analysis that qualify for conclusions about deeper nodes and backbones. This is a tree that we have made in order to look at the end tips, use phylogenetic methods to sort out the lineages and to look at the end tips that supposedly should represent a species. That would be nice. If we do this on conserved genes or on ITS, we will be rather confused, but the combination of heat shock protein and RPB2, which give just an alignment, that's what we can call barcode length, 620 base pairs, that tells us a lot. And I'm not operating at this stage in our work with clades because of bad support of the deeper nodes. I'm using species group. And we are talking about a group of species where we have found, according to 79 lineages, and if you ask me, some of you, how many Helvella occur on the globe, I think that we at least have to multiply it with 2 when we get materials from other parts of the world. So we have also we have made similar phylograms with 3 genes, with 4 genes, but of course then in order to show you the 4-gene tree, then we would have to exclude a lot of specimens and species, because these barcodes and the primers, they don't function on all the nice specimens that we've gathered together. So we present now the 2-gene tree. Here is one of those that you can see in the tree. They have got blue. It's difficult to see for you. There are 3 species that have been combined in Helvella and participated in creating a rather broad Helvella generic border, and that is Helvella estivalis, which you can see up to the left there in the phylogram. It's Helvella sylvicula, more commonly known as Vinella sylvicula, which also are up in the left corner. And then we have a 3rd species called Pindara terrestris or Helvella terrestris, because we, Sara Landvik and me, we combined it in Helvella in the late 90s, based on some molecular work at that time. But now we have surveyed it in relation to our broad sampling of Helvella from Europe. I just show a picture of these nice fungi. This is Helvella estivalis, which is very common in calcareous soil in the Arctic and Alpine zones, described from France from the beginning. And there you have this Helvella or Vinella sylvicula that was combined in Helvella by the Finnish mycologist Harmaja. And there you have these very globally very rare, and I haven't seen it in the field myself, but Seppo Huhtinen says that he has seen it in Finland. We have 3, 4, 5 collections from the Nordic countries. And so let's now go into some part of the phylogram. And the important thing is that the combination of these two barcodes, when we screen material from different parts of the world, I think we've succeeded to find a combination of genes that separate good on species level, and still there are very genetic consistent between specimens coming from different parts of the globe. And here you can see what we call the acetabulum complex, which in fact has lived and still are identified mostly as acetabulum in the fungarium around the world, where you can see very, very distinct species. And especially the Finnish mycologist Harri Harmaja, he was particularly insightful when he, in the 70s, described these new species, Arctoalpina and Dryadophila. And he described those two species from Norwegian material, and we have included the holotype specimen in our analysis. So we know definitely what this is. It's also interesting for all those from America, but maybe I don't have time for this, but Costata was also one of those species that was adopted by Fries. It's an Albertinian Schweinitz species from USA, and this is a collection, Costata, you can see there, from California that we got from Harvard, and definitely what has been called acetabulum in America should be redescribed and neotypified, but that work we leave to others, colleagues, to do. We concentrate now on the situation in Europe. Here is Helvella Pezizomycete, which is the name we have adopted. A lot of confusion in relation to the naming of this, but it's beautiful. That's why I show it to you. And Helvella acetabulum, be aware that it can look like this. There are morphological variants inside. Even those species that the molecule tells us are good genetic differentiated. Here is one of those without the stem, but you can also find them with a more elongate stem up. And then it's almost impossible to discriminate from another new species who's called Helvella hyperborea. So morphology may vary within a species, and it's very hard to identify because, as is concluded in the abstract, the microanatomy is we don't have many characters, and taxonomists will call it subtle characters. And again, the message is that morphology is a very bad predictor of phylogeny and to be used in classification, and that certainly applies for Helvella. Here you have these growing with Dryas. You can see some leaf of Dryas to the left in the slide, Helvella dryadophila, which is a good species. And this Arctoalpina that was believed to be an acetabulum, which was also an Dryas ectomycorrhizal. In fact, this ectomycorrhizal relation has been proven in a master thesis at the University of Oslo, but not commonly referred to, but that was done in '98. Yeah, now we go to another group that has caused a lot of trouble. In a recent work from North America, it was concluded based on LSU analysis that what people had called lacunosa in North America included at least 12 different species. I don't fully agree with that conclusion, but nevertheless, it tells that the molecular variation, because that was a pure molecular study, then it's great within some of the subclades, but in other subclades, the evolution is much more retarded, and it's very difficult to use molecules alone, even those good barcode molecules in combination, to separate, discriminate among species who are definitely morphologically divergent. I will give you an example of that. In this Lacunosa group, to the Mediterranean people here, you also see we have a very long branch there going to some Mediterranean taxa from South France, Canary Islands, and Portugal. But I don't have time to enter that stuff now. Just show you some of the beauties. Helvella flebophora, I think, is the most beautiful fungus out there, especially if you turn it around and you see these nice ribs and wheel on the underside of the apothecium. The white Helvella is very problematic, and a part of the work has been to rename and resurrect name, which I think a taxonomist should do. And the easy way is, of course, to describe things as new and forget everything pre-Frisian and what earlier morphologists have done. But when we know that they have operated with broad species concepts, and a lot has been synonymized, but maybe that was not the best solution to do, then we have to go back and try to find proper names for these. And in fact, we have resurrected 12 nominals from also pre-Frisian work. I hope you all, when you work on these groups, not only ASCOs, but are aware of Jakob Christian Scheffer's important work and iconographs, and that was in 2 parts published. First, not with binomials in the 1763, and then later on he put binomials to all his taxa. And Fries, he sanctioned many of those names as synonym on species level, but mostly he included them as an alpha or beta, a forma, a variety, and sanctioned on a different level, which is, so these names, they are still useful. And yeah, so there you have this important work that I would encourage you to take a look into. Yeah. Here you can, this is my example on how important it is to have been out in the field and to get knowledge about these fungi. And I will use an example of a species I described myself a couple of years ago. I called it Helvella daurensis because at that time I had only seen it in the Norwegian mountains. And look at the apothecia, how they look like. And if we go then into this part of the phylogram, you can see that there are daurensis there. I found it in the French, Swiss, and Austrian Alps. But there is another species also called palustris. And the differences between those there are only a couple of base pairs. But it looks like it's much more because we have enlarged the branches. But it's only, so a molecular systematism would definitely, based on this and a general overview, put palustris and daurensis in the same group. But this is palustris, which is quite different. You have it in, you also have it in North America. It was described by Peck. But this has a very, very distinct ecology, which is definitely not growing in places where you have daurensis. But the molecules that do not tell us which species do we have, but morphology they do. Okay, so the time is going, and another group which is very problematic and related to what we have seen, it's important when we give things to these ecologists and so, that we apply a name that means something. And Helvella elastica, you will never know what you give to an ecologist or to a survey on species on higher level when you pick an elastica out from the herbaria, because there are so many species there that we have now resolved and put old names on. And Yeah. And here is the last one, which is also a little problematic taxonomically. It occurs in western part of US, together with Europe, but in the western part you have one called Stevensy. So at the very end, I would just like to acknowledge the late Dr. Henry Dissing. It's a picture I found for taking me out in the field when I was a young student and teach me about Helvella. And of course, I will also acknowledge those two in the mycology group who has worked together with me on these Helvella work. Thank you.